but I will talk about some human work as well. This is just a picture of, that is just a picture, for those of you who have not been to Portland, this is the Portland VA, which is connected to Oregon Health and Science University by this long sky bridge. So I just want to briefly go over my disclosures. So Oregon Health and Science University, myself, and some other investigators have a significant financial interest in RTL Immunotherapeutics, which is a company that may have a commercial interest in some of the research that I'm going to be talking about today. And these potential individual and institutional conflicts are being managed by the committees at Oregon Health and Science University. Okay, just a few caveats in terms of research. So I'm sure we all are aware of this, but it's just nice to be reminded before we dig into some studies. So they vary obviously in terms of their quality, in particular their sampling methods, so their choice of subjects, whether they're adults or male or female or rodents. Also inclusion and exclusion criteria, so the duration of abstinence, how long they've been addicted to the substance, and of course the presence of comorbidities. So in animal models, it's very easy to study alcohol abuse only, but in humans, it's a lot more complex. Also, there's variability in the outcome measures that are selected. So when you're looking at assessing mood or cognitive function, for example, there's self-report measures versus more functional assessments. And then a big question of how polysubstance use is addressed. So like I mentioned with animal models, you can be a lot more controlled, but with humans it's a little more challenging. Okay, so those are the limitations. This is just an overview of what we're going to go over today. So I'll start with a little background and also talk about some targets in the immune system that are being developed for therapeutic intervention. And then I will go over a few different immunomodulatory treatment strategies, and then some future directions. So background. I show this oil painting to really kind of illustrate the link between our immune system and our mental health. So if you weren't seeing the title of this art, you might think that she's also just potentially sick. Or depressed, it's unclear. So there's this kind of fine line, and sort of I think within this delicate balance, some opportunity for intervention. Okay, so my laboratory is focused on, this is a long word, translational psychoneuroimmunology, which is really translational, meaning just from the lab to the clinic, but the psychoneuroimmunology piece, because I was trained as a behavioral neuroscientist, I also went to graduate school for psychology, and then I did a postdoc in immunology. And so I'm not creating this word. This is already kind of a subfield, but it really, I think we were talking about a holistic approach, and I think for addiction that's especially important. And so we study mechanisms by which substance use disorders will induce abnormalities in immune cell function and contribute to neuropsychiatric impairments. And so the key questions that we're looking at in the lab are how methamphetamine affects immune cell function, and in particular T cells and these glial cells in the brain, and then how do changes in immune function go to influence brain and behavior, in particular psychiatric impairments that will impede recovery. And so why do we want to target the immune system for substance use disorders? Increasingly, preclinical and clinical studies are showing that substances of abuse can have significant and sometimes long-lasting effects on the immune system, which then can influence behavior. This is the National Institutes on Drug Abuse kind of top 10 drugs that are abused, and today, and the point is that almost all of those you can find evidence in the literature impact immune function in some way. And today we're going to focus just on the stimulants, and in particular methamphetamine. Okay, so this idea that the immune system can influence behavior is not new, but I think you can adapt it. Substance use can kind of fit in with this story. So this is a slide that was from Robert Dancer's group, published several years ago from a cancer perspective, but showing that chronic inflammation, exogenous cytokines, so cytokines that are taken for therapeutic reasons, cancer and stress can all influence peripheral cytokines, which then affect your brain and can cause various neuropsychiatric impairments. So anhedonia or depression, cognitive alterations, fatigue, depressed mood, etc. And so with substance use disorders, you also see chronic inflammation and stress, which fits right in with this model. Here's some examples of some cytokines that you might have heard about or be familiar with. So there's the interferon. So interferon alpha is used as a therapeutic for cancer as well as hep C, although that's changing a bit. Tumor necrosis factor alpha and beta. There's the interleukins. There's a lot more than 10. I'm just showing a few key ones here that people talk about sometimes. Macrophage migration inhibitory factor, or MIF, is a cytokine that I'm going to be talking about later because this is one of the inflammatory factors that we think our therapy helps kind of mitigate. And also monocyte chemoattractive protein, or MCP1. And so I know we're all aware that methamphetamine use is associated with serious physical and mental health disorders. We are particularly interested in looking at kind of the cognitive impairments as well as the mood impairments, so depression. And we think that impaired immune system kind of plays a role in contributing to some of the damage that is occurring in methamphetamine. So this is an example of a study that was published in the Journal of Neuroscience back in 2004, but I like to bring it up because it illustrates some interesting points. So this was a study of 22 methamphetamine addicts who had been using methamphetamine for a number of years, at least 10 years, compared to 20 matched controls. And they did some imaging, and they found that the limbic region of the brain, which is involved in reward and mood and craving, lost about 11% of its tissue. And the hippocampus, which is a region of the brain important for cognitive function, lost about 8% of its tissue. And that, they said in the article, is comparable to the deficits that you might see in early Alzheimer's disease. And the red areas here are showing kind of the areas of the greatest tissue damage. But one thing that the authors noted that wasn't highlighted in the paper as much, but they talked about in a press release following this study, was that a surprising finding was that the brains of the meth addicts were roughly 10% larger than normal. So, there we go, growing brain. They were surprised by this finding, but encouraged at the same time, because that meant that the tissue wasn't dead or damaged, that it was inflamed, but that it could heal. And so another study kind of with similar findings, also looking at some human brain imaging. This was looking at dopamine transporters in the striatum and in protracted abstinence. And what they found here, so these are a normal control brain, and here's a meth user 1 month after abstinence, and you can see that the loss of the dopamine transporter is apparent here because there's not the red. The red is showing kind of high dopamine transporter levels. At 14 months after no meth use, you see a restoration of the dopamine terminals here in the striatum, and that's encouraging, but what they didn't also find was that in these same participants, they didn't see an improvement in neuropsychiatric function, suggesting that the brain can recover, but dopamine is probably not the only story or only answer here, and so there must be something else going on. Okay, so, and maybe it's immune related, and so I'm going to talk now about some targets for development that are kind of focused on the immune system. And so glial cells are one candidate here, and I'm just going to highlight, let's see here. Yep, okay, so glia, they recently have been becoming more and more studied, but historically neurons were kind of the cell in the brain that people were most focused on, but glia actually are more prevalent than neurons. They have many of the same receptors as neurons. They secrete neurotransmitters and other inflammatory factors, and they also can play a key role in synaptic plasticity. So the microglia the glia that we're going to talk about mostly today are astrocytes and microglial cells. And so take a little closer look at those cell types here and how they relate to stimulant abuse. So this is a kind of a complicated image, and I'm just going to highlight a few aspects. This is a kind of a schematic showing how psychostimulants may impact these different glial cells and also contribute to kind of maintaining neuronal damage. And so psychostimulants will create reactive oxygen species or free radicals, which then will induce these DAMPs or damage-associated pattern molecules. And so those are then recognized by the glial cells. So there's receptors for these damage signals on the glial cells, and then in response to the damage signals, the glial cells will then secrete pro-inflammatory factors. Like we see here, we talked about tumor necrosis factor alpha, we see here interleukin-6, so kind of creating this positive feedback loop. So neuronal damage plus a heightened state of glial activation will result in positive microglial and astrocyte and neuronal and glial feedback, which then can kind of increase neuroinflammation and injury in this sort of self-perpetuating mechanism. And so the idea is to try to arrest this and allow healing to happen. So our work is focused on kind of another target, looking at myelin, the sheath that surrounds the axons of the neurons, and the role of T cells. And this therapy was originally designed to treat multiple sclerosis, and it's been used in animal models of stroke. And what they've found in those studies is that these T cell receptor ligands will inhibit recruitment of inflammatory cells to the brain. They can help repair myelin and axonal damage, and they will also help restore impaired motor function in, in animal models of multiple sclerosis. And so the molecule that we are studying is this RTL. It's comprised of, for you, chemists in the room, of a beta chain, which is in blue, and the alpha chain in red, and then you have a target peptide here in green. And our target peptide is myelin oligodendrocyte glycoprotein, or MOG, which is potentially a target for MS. And here's kind of another view of how we think the drug works. So RTL-1000 is the drug that is used in humans. RTL-551 is the drug same drug, but it's the mouse version. And so what we think that RTLs do is bind to CD74, which is the receptor for MIF, one of those pro-inflammatory cytokines. And so by blocking the access of MIF to its receptor, you then are able to reduce the number of cytokines, you increase apoptosis of autoreactive T cells potentially, and then you increase random migration so that you don't get kind of this trafficking and clustering of inflammatory cells. And we think that those are mechanisms are kind of going to contribute to RTL's therapeutic effects in methamphetamine and potentially other substances of abuse as well. And here's some just early unpublished data that we've found in our adults, humans. So we have control participants. These are only about eight per group, but we have active methamphetamine users and those in remission, and we find that the participants in remission actually show higher levels of this cytokine, suggesting that this may be an effective strategy in adults. Okay, so shifting gears now, we've talked about some of the targets. I'll talk about how these targets are working in the laboratory. This is just to remind us all that substance abuse is very similar to other chronic conditions where 40 to 60% of relapse is common. This should just really be a trigger for renewed intervention and not something to suggest that they've failed in some way. So, and we are focused primarily on kind of how to help reduce relapse, but at the same time acknowledging that it may be that you need to treat some of these neuropsychiatric impairments to really help that happen. And so just as an example of that, in our, this is a kind of a translational study, so these are mice, and let me get the rest of this up here for a second. Okay, so these are mice in early recovery, in late recovery, and then this is our saline control group. And then these are results from our human participants, and we can see that this the cytokine MCP1, this pro-inflammatory factor, is elevated in late recovery for the mice, and this is a graph showing days of remission from methamphetamine. So you can see that MCP levels tend to go up during remission in these adults. At the same time, these same participants also reported more memory problems, and they performed worse on tests of attention and executive function, whereas we didn't see those cognitive impairments in the active meth users. Okay, so some of the therapies that are under investigation are antibodies to attract substances so that it's too large to pass through the blood-brain barrier, which is vaccine strategies, and that's a very large topic that I'm not going to cover today, but I can certainly provide references if you want to read more about that, but those are some potentially promising approaches. There's also a strategy where you can suppress glial cells by using ibudilast, and it's a phosphodiesterase inhibitor, and I'll talk about some of their preclinical work and kind of let you know where that drug is in terms of its clinical development. There's also, this is our approach here, to kind of reduce the damage that you see from methamphetamine, allow the brain to heal, and improve neuropsychiatric function. And finally, I didn't want to focus only on kind of drug approaches because I think, again, as we heard this morning, that kind of a holistic approach is ideal, and there has been some work just looking at exercise and how that can really improve recovery, and so I'm going to speed up here. So this is kind of the human piece I'll talk about in a minute. Okay, just quickly to show what this looks like in an animal model. This is the ibudilast I was mentioning, and these are just different drug doses. But if you just want to focus on this lower bar, these are showing how many times a rat will press a lever to receive an infusion of drug. And you can see that this dose of ibudilast was pretty effective at reducing the number of lever presses, so drug seeking seems to be decreased. And this is an example of stress-induced reinstatement. So these rats were previously trained to lever press for the drug, and then that behavior was extinguished, and then it was reinstated by using a stressor. In this case, it was a foot shock. So you can see that both these doses of ibudilast were able to kind of decrease the relapse that may occur following a stressor. So preclinically, it appears that ibudilast will cross the blood-brain barrier. It's well tolerated. You can give it orally, and it does reduce glial activation. In other work, this is done in human in vitro cells. These phosphodiesterases have also been shown to inhibit tumor necrosis factor alpha, which again is another pro-inflammatory factor which may contribute to these impairments. In addition to looking at kind of drug-seeking behavior and relapse, you can also look at cognitive functioning, and so that is one of the things that we do, and I will just kind of briefly describe the tasks for those who are not familiar. This is the Morse water maze. It's a tank of water that's divided into 4 quadrants. The animal is given 60 seconds to swim and find the platform, and there's different training sessions. So there's a visible training session where there's this little sort of beacon on the platform, and then there's hidden platform training sessions where we can see it because we have this aerial view, but the mouse in the water can't see that. It's submerged a few millimeters below the surface of the water. And then on the last kind of test day is a probe trial where you record the amount of time that the animal spends in the quadrant where the platform was. And this is what that looks like. So here's our treatment groups. We have our saline and vehicle controls. These are the meth-only group. Some more controls, and these are the meth given the RTL. And you can see that the goal here is the black bar is the target quadrant. And so you can see that with the meth-treated animals, they didn't show any kind of preference for that quadrant where they thought the platform was. And then over here, I'm just showing you that the RTL was able to improve their ability to find the quadrant. We did this while they were concurrently receiving methamphetamine, as well as in a model where they had a period of remission from drug use. So kind of in both these models, it was effective. And I know I'm out of time here, so I will just quickly show you that we also found that the RTL was able to reduce some inflammation in the brain, in the hypothalamus in particular. And this is the exercise intervention I was mentioning. So 8-week intervention, 135 participants. The bottom line is that exercise had a significant effect on reducing depression as well as anxiety. And so results suggest that exercise may be effective at improving symptoms of depression. and as well as inflammation. Okay, I'll skip the summary and just tell you what's going on with these trials. So researchers have found that the anti-methamphetamine vaccine is safe, and that's as far as they've gotten with that so far. For ibudilast, they've completed a phase 1 trial, and a phase 2 trial is currently underway. They estimate being completed in June of 2017. We're working on some other immunotherapies, and we're trying also to get a trial started for our RTL in adults with methamphetamine addiction. Okay, and I just want to thank my collaborators, and here's kind of another picture of Portland for you.